Good day everyone and welcome to the discussion of the citric acid cycle. What is the citric acid cycle? By definition, the citric acid cycle is the series of biochemical reactions in which the acetyl portion of acetyl-CoA is oxidized to carbon dioxide and the reduced coenzymes FADH2 and NADH are produced. In other words, it is a biochemical pathway that is used to generate energy to the oxidation of acetyl-CoA and is used for the synthesis of NADH and the production of amino acids. The citric acid cycle is the central metabolic hub of the cell. It is also known as Krebs cycle after its discoverer, Hans Adolf Krebs, and as the tricarboxylic acid cycle in reference to the three carboxylate groups present in citric acid. It is a common pathway for the oxidation of fuel molecules such as amino acids, fatty acids, and carbohydrates. In eukaryotes, the reactions of the citric acid cycle take place inside mitochondria, in contrast with those of glycolysis which take place in the cytosol. The first step involved is actually a preparatory step which begins with pyruvate. Pyruvate is derived through the glycolysis of glucose, which is 6-carbon compound, and it is split into two molecules of pyruvate, which is a 3-compound carbon. The next step includes the oxidation of pyruvate into acetyl-CoA by the enzyme pyruvate dehydrogenase complex, and in this reaction, a molecule of NADH is generated. The acetyl-CoA is a 2-carbon compound. In the next step, the acetyl-CoA combines with oxaloacetate, which is a 4-carbon compound, to form citrate, and hence the resulting molecule is a 6-carbon compound. This reaction is catalyzed by the enzyme citrate synthase. In the next step, the citrate is isomerized into isocitrate by the enzyme aconitase. Next, the isocitrate is oxidized into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase, and in this reaction, a molecule of NAD is reduced to NADH and a molecule of carbon dioxide is generated. Hence, the alpha-ketoglutarate is the 5-carbon compound since one molecule of carbon is lost into carbon dioxide. Next, the alpha-ketoglutarate is converted into 16-alkyl by the enzyme alpha-ketoglutarate dehydrogenase, and in this reaction, also a molecule of NAD is reduced to NADH and a molecule of carbon dioxide is released. Hence, the succinyl-CoA is the 4-carbon compound. In the next step, the succinyl-CoA is converted into succinate by the enzyme succinyl-CoA synthase. In this reaction, a molecule of GTP is generated. Next, the succinate is converted into fumarate by the enzyme succinate dehydrogenase. In this reaction, a molecule of QH2 is generated, which is used for the production of FADH2. Fumarate is then converted into malate by the enzyme fumarase. In the last step, the malate is converted into oxaloacetate by the enzyme malate dehydrogenase, and in this reaction, also NAD is reduced to NADH. Let's look at the result of Krebs cycle. Through each cycle of the Krebs cycle, the following products are generated: 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of GTP, and 2 molecules of carbon dioxide. Now, since glucose is split into two pyruvate compounds, for each molecule of glucose, this cycle runs twice. Hence, the products formed are 6 NADH, 2 FADH2, 2 GTP, and 4 carbon dioxide. All the NADH and FADH2 are next fed to the electron transport chain for the generation of ATP. And that's all for the discussion of the citric acid cycle. Thank you for listening.